Today we're going to be emphasizing what happens when DNA becomes mutated and how that might have impacts on our proteins. But first of all, let's review a few things about the cell in relation to proteins. So we should know that all of the genetic instructions of a cell are in the nucleus. So in the nucleus, I added a little extra detail. Remember, there is a nuclear membrane around the genetic material. There are pores in that nuclear membrane also that will allow materials to enter and exit the nucleus. So if I were to open up the nucleus, inside of the nucleus, we would expect to find genetic material or DNA. There would be very long strands of DNA. And we know that this DNA is made up of adenines, thymines, guanines, and cytosines. Now, this molecule is way too valuable to be allowed to leave the nucleus for any reason, especially to follow the instructions in terms of making a protein. So from that genetic material, our cell will actually construct a message, and that message is called messenger RNA. So one of the things that we've already learned is how to build a strand of messenger RNA from a gene on the DNA or of the DNA. So we might transcribe and make a message or messenger RNA from this particular stretch of DNA. So this is our messenger RNA, and that messenger RNA will leave the DNA through one of the pores. So we'll take it out through maybe that pore right there. And that messenger RNA, when it makes it to the cytoplasm, is going to be able to then be translated into a protein. So again, the first step in building a protein is called transcription. And transcription is the process of taking a molecule of DNA and rewriting it into a piece of messenger RNA. Now that piece of messenger RNA, once it arrives or gets to a ribosome, then needs to be translated. Now the ribosome looks like a very small circular type organelle in the cytoplasm. We often draw them as dots, but in reality, the ribosome is actually a piece of RNA itself that is coiled into a large subunit and a small subunit. So if we take that messenger RNA that had left the nucleus for a ribosome, the messenger RNA will travel between the two subunits of the ribosome in order for translation to then occur. So we've now taken the messenger RNA. Your ribosome will decode or read that message in order to build a chain of amino acids. The order of those amino acids will dictate the final three-dimensional shape, as well as then the protein itself, which will have a particular purpose for the cell. That purpose may be as an enzyme, it may be as a particular protein in the cell membrane. Perhaps it is a pigment that will give you your eye color or your hair color. Perhaps it's even a molecule in your muscles that will help to develop dimples. That's the purpose of proteins. So every gene in your genome or every section of your genetic material does have a purpose, and the protein or instructions from that DNA will be translated by the ribosome. Let's start with a basic definition of mutation. A mutation is going to be a heritable change in genetic information. That means that it can be inherited or passed from one generation to the next. So this would be a change that occurred in the DNA that can then be passed on to the offspring. So, as you can see, we have a segment of DNA. Now keep in mind, if this is one side of the DNA, that means, if I draw back a little bit, that this DNA has been untwisted or uncoiled and it has been unzipped. So there is a second side or strand to the DNA, but that is not the side or strand that we are going to transcribe. So instead, I have a stretch of DNA, and when I go from DNA to messenger RNA, that will be our first of 2 steps. So from DNA to RNA is step 1, and we know that step 1 is called transcription. This is not the first time you've done this, so I'm not going to belabor the steps. So if I would transcribe this particular piece of DNA, I'm going to end up with AUG, CGU, ACC, UUU. So I used our base pairing rules to build our messenger RNA, making sure to watch out that I use uracil instead of thymine. So that was step 1, and that was our transcription. Step 2, which would occur in the ribosome. So the messenger RNA has to move off to the ribosome. So we go to step 2 to build our amino acid. Step 2 we know to be translation. In translation, we need to use our tools, whether you prefer the round or the square. And we again know that the ribosome is going to read these in 3s or a codon, starting with an AUG. So I started us off with an AUG, so not trying to be tricky. And then when I start to use the chart, I can see that AUG is going to be methionine, as should all proteins should start. CGU is arginine. Your ribosome will bond those 2 together. After arginine, ACC, I get threonine. And lastly, UUU, I get phenylalanine. Now, notice that we did not get a stop. That tells us that this DNA and messenger RNA must have continued. Most proteins are very long, so that would mean our amino acid chain would technically continue also, but we're going to concentrate on these 4 amino acids. We're going to consider this then our normal protein. So when we look at future mutations, as we mutate the DNA, we want to see if there is any resulting impact on the protein. So as we go to our second example, please take a minute to write this one down. As we look at the second example, again, I'm going to transcribe my DNA. As I look at my transcription, there is a difference. So I'm going to go back. I'm going to take a step back. I'm going to go look for the difference in the DNA. If I go from one base to the next, comparing my original to my mutation, I'm looking from one base of the next to the next of my DNA. One base to the next. It looks like we have a change in our DNA here in the second codon. Instead of a C, it looks as though we now have a T. If I look at the rest of our strand, everything else does look to be the same. So essentially, we have a base of a T that has been put in place of or has been substituted for a cytosine. So we need to see if this has any impending changes to the protein. So again, I'm going to start with an AUG, so we're all good there. As we translate, we know we're going to get a methionine. And here's the codon that had our change, had our mutation. So when I look now and I have a CAU, I do get an amino acid of histidine. ACC, threonine, and lastly phenylalanine. So we do see from our substitution that we did get a different base. So that's precisely what we call this type of mutation. It is called a substitution. And I think that name makes a lot of sense. Now, as we go to look at our next example, we need to be cautious because as we start comparing, we are still going to go back and compare to our normal. As we look at our next example and knowing that we are looking for mutations, let's start out looking from DNA to DNA. So if I go base by base, I look for changes. I'm not seeing any yet. Oh, there we go. So here we do have a change to our DNA. Rather than having a guanine in normal, we have a cytosine in our mutation. The last three bases all look to be the same. So again, we do have a substitution where we have, let me make sure I get the right G. A cytosine has been brought in replace of the guanine. So we do again have a substitution mutation. So in my notes to the side, I've made a note that it's a substitution. All right, if we do our thing, we transcribe it. We translate it. Now, as far as the mutation is concerned, if we compare our normal protein to our protein after the mutation, MET, ARG, THR, and Phi. So although we know we had a change to the DNA, we definitely have a mutation, there is no resulting impact on the protein. And if you look at those amino acid charts, there are numerous situations where you can get the same amino acid from varied or different codons. So this is still a substitution. There is a substitution mutation. However, because there is no change to the protein, this is also referenced or considered to be a silent mutation. And again, I think that name makes a lot of sense. Now, in both cases, in our two substitutions, there was only one point in the DNA where there was a mutation or where the DNA changed. So we can even call this, we can group this together as what is called a point mutation. There is one distinct location that was impacted. Now, it doesn't have to be just one base. It could be 2 bases or 3 bases, but if it is just that location, we consider that a point mutation. I can point to the change in the DNA, and that is where I expect the change in my amino acid or protein to be. A substitution is not the only kind of mutation that we want to observe. So we are again going to be comparing to that same normal gene, and that gene has 4 amino acids that we've identified. So as we look at the new DNA, this DNA is actually still identical to the normal. It has not yet been mutated. So we are going to actually perform a mutation called an insertion. So over here to the side, I'm going to title this one an insertion. So we are going to insert a base, and I'm just going to randomly choose. I want to insert a base right here, and I'm going to insert a T. That is totally random. I have no idea what our outcome is going to be. Please take a minute, transcribe and translate this piece of DNA with the new mutation. Based on the location of our mutation, we would imagine that our mutated DNA might impact this second amino acid, seeing as it seems to be right around the second codon. So the fact that when we look at a normal or original protein compared to our new protein, it's kind of interesting. We still ended up with the same arginine, So the mutation did not occur exactly where we expected. However, we do have changes moving down the amino acid chain. So this mutation actually did something a little different. If you notice, you even have an extra base. Now, we've already said that we expect that this protein continues, so we would expect that that U would become part of the next codon, but that's definitely different than what we saw in our normal DNA. So really what happened is by making that insertion, it shifted all of the codons. So even though our arginine, by chance, remained the same, because we added a base, it shifted all of our codons. In our next example, we are going to do a deletion. You might have expected that. And again, I'm going to randomly choose a base to delete. So I'm going to go with, I don't know, this G right here. I'm going to delete that G. Please write down that DNA sequence, transcribe it, and translate it. So again, if we look at where our deletion occurred, we would expect perhaps this third amino acid to be the start of our mutated or changed protein. We very randomly ended up with the same first three amino acids as our normal. Remember, we have to go all the way back compared to our original normal. Put that right here in place next to it so we can compare them side by side. So it is kind of interesting. I mean, that was totally random. I just picked a random base. So we do, just per chance, end up with the first three amino acids, but we've actually reduced our amino acid chain, it seems. We only have two bases here at the end, so we cannot figure out the next amino acid. Again, knowing that we expect this DNA and this mRNA would have been longer. We've never had a stop codon in all of this. So this UU would now lead into the next codon and very likely change the future amino acids that were part of this particular protein. So we did randomly end up with not a whole lot of change right now, but because we have one fewer amino acid, we can still see that we have shifted these bases because of that deletion. So with that in mind, we look at an insertion and a deletion. We can group those together in what we call a frame shift mutation. And again, a frame shift mutation is a result of shifting or offsetting those triplet codons. And we can see some evidence there by noticing that we have a leftover nucleic acid here, base that is, and here we have two but do not have a third to fill that codon. So these are really the major types of mutations. There is one last example to show you. I'm not going to have you write out this particular DNA strand. It's actually a pretty lengthy one, but I will kind of go through this for you quickly. So DNA to messenger RNA, we know that's transcription, to throw out some vocabulary for you again. So if I quickly transcribe this particular strand, you're probably getting about that fast at it also. I know I need to find an AUG, so here it is. There's my start codon. So 1, 2, 3, 4, 5, 6, 7. Looks as though I'm going to have 7 amino acids, presuming that if I transcribed all of this, it'll be a part of my protein, so probably at least 7 amino acids. I know by now, I've done it enough, that AUG is going to give me a methionine for my first amino acid. When I look at my next amino acid, UAG, I get a stop already. Now, if you can tell, I actually made a change and mutated the DNA right here. I made a substitution change, And when I created that mutation and I changed the DNA, it caused my protein to stop prematurely. My stop really would have been way down here. So if I actually transcribed this and translated the entire thing, my stop codon would have been way down here toward the end. So instead, this stop has happened early. So this is one more type of mutation called a nonsense mutation. And it simply means that the protein stops, because we had a stop codon, the protein stops prematurely or early. So the protein really should not have been 1 amino acid long. In this case, like I said, the stop would actually have been down here at the end. It would have been 1, 2, 3, 4, 5 amino acids long. So because of that substitution mutation, we ended up with a nonsense protein. So we call this a nonsense mutation. It's time for you to try a few examples on your own. So we're going to go back to our color-coded genes. It turns out that these genes are actually the genes of scorpion. And the first particular example that we have is for the color of our scorpion. So our normal scorpion is actually going to be black in color. Most scorpions do tend to have very dark colors to them. So this particular gene does contain the instructions to result in a black scorpion. Please take a minute, transcribe and translate so that you can determine which protein is going to give us our scorpion color. When we go ahead and transcribe and translate for our protein, please don't forget that you do need to find an AUG as your start codon. So we have 4 amino acids in this particular protein. I guess I should point out all of this is fictitious. These are all just DNA codes that I created so that I could create this little activity. So we have methionine, aspartic acid, phenylalanine, and lysine. Lastly, we have a stop. So what we are going to see now is comparatively, I have a piece of mutated DNA. So there is something different about this particular piece of DNA. Now, just so that we don't have as many letters to look at, I'm going to go ahead and use my helicase and cut open or separate, unzip our DNA. So the first suggestion is that you go down through and you actually find the mutation. Where is the DNA different? Because I've definitely mutated the DNA somewhere. So if I start and look at my bases, A, T, 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 A, C, C, T, A, T, T, there it is. I have a C on my normal DNA strand, but a G on my mutated strand. So I have a mutation right here. So that looks to be something like a substitution, but you're going to have to find out if the protein changes. If the protein doesn't change, then it's a silent mutation. If the protein stops early, it's a nonsense mutation. So we need to figure out what type of mutation this happens to be. Please transcribe and translate your mutated DNA now. So when we compare our 2 proteins, we should know exactly where to expect a potential change in our protein. That shouldn't happen until we get down to the mutation itself. So I still have methionine, aspartic acid, phenylalanine, and where I had lysine before, I now have asparagine. So I do have a change to my protein. Now, right now, we don't know what kind of impact that is going to have on our scorpion, but we do know that if this mutation occurs, our scorpion will not be black. We will save that information for tomorrow, but for now, our scorpion will not be black because its DNA has been mutated. Our second trait is going to come from a gene for legs. So again, this is to determine what type of legs our scorpion is going to have. Now, scorpions are arthropods. So scorpions have jointed legs, and they have 4 pair of them. So our normal DNA is to determine the normal jointed legs of the scorpion. You also, though, have your mutated DNA, so I will help you out. Helicase will do its job. Now your DNA is ready to be transcribed and translated. When the video pauses, please write down your DNA strands. You're going to want to look for where the mutation in the DNA looks to occur, and then check to see if the protein changes as well. When transcribing and translating the DNA, I'm starting to find some shortcuts. You know, I know that I need to find a UAG to get me started, so I didn't even transcribe or translate the beginning. I need to find an AUG. So I found an AUG and worked out to have methionine, valine, and lysine before I got to my stop codon. In the mutated DNA, though, as I was working and I blocked off all of my codons, I noticed I have an extra base. So when I have an extra base, that tells me that there must have been an insertion. If I go back and look at my 2 stretches of DNA, I will be able to find the extra base. So my methionine was the same, valine was the same, lysine was the same, but I did not get a stop. So my insertion was here in this 4th amino acid, and now that tells us that this protein is actually going to continue when it should not. So we are not going to have 4 pair of jointed legs for this particular scorpion. You will find out in the future what that mutation is actually going to do to our trait, but we will not have 4 pair of jointed legs. Your scorpion will be different. Here's a gene from the scorpion, and this gene codes for its pedipalps. You might want to call them the pinchers or the claws. So same process. We don't know what the mutation is going to potentially do to these pedipalps, but we're going to figure it out between today and tomorrow. So helicase unzips the DNA. I would like you to compare your 2 strands of DNA, see if you can find where the mutation occurs, looking 1 base at a time. Finally, you want to transcribe and translate to see if you have a change in the protein. When I look at the results from my pedipalp investigation, my normal protein met arg sur tri arg, and then it stopped. I found my mutation here, looks to be a substitution, but let's make sure that the protein is actually changed. I don't expect my protein to be different until I reach my mutation, so that makes the beginning part pretty quick. It turns out, though, that even with the mutation, I still got the same amino acid. So this means that here we have a silent mutation. Although there was a mutation to the DNA, here we have a silent mutation. So that means that we would actually not see a change to this particular scorpion's pedipalps. Our last trait in regards to the scorpion is with its venom. So you likely know that a scorpion uses its venom to stun, paralyze, or kill its prey. And most scorpions have a powerful enough venom to kill things that are smaller than it is. So in terms of this particular scorpion, we're going to assume that its venom is of average strength. It's not going to disturb or really harm you or I. It might kill some skin cells, but it is not going to be poisonous or deadly. So as we look at our DNA, I'm going to ask you to disregard that circle. I was looking at the wrong thing. I had my DNA flipped backwards. If I compare my DNA, I'm going to unzip it just so I have fewer bases to look at and compare, and we're going to find our mutation. If I compare each set of bases as I look from one to the other, G, C, C, T, G, A, T, A, C, A, here's my mutation. Having trouble circling. There we go. C, A, A. So there is our mutation. That will be the approximate location that we might expect to see a possible change in the protein. We'll try to figure out if this was a frame shift or a substitution once we start transcribing and translating it.